Hello, everybody. In today's discussion, we're going to start looking at molecular analysis. This would go along with Chapter 12 in the McMurray Organic Chemistry textbook. And we are going to start with a discussion of mass spectrometry. So as we can see on this initial slide, there are four primary types of organic molecular analysis that we would do. Mass spectrometry is one. This is often done in a core facility. Some individual labs might have a mass spectrometer as technology evolves. Later on, we'll talk about infrared spectroscopy. This is done in every organic lab everywhere. Nuclear magnetic resonance, or NMR spectroscopy, and ultraviolet spectroscopy. Somewhat more limited usage, but all four of these are important in having this comprehensive picture of what is involved in an organic molecule structure. So mass spec in particular will tell us something about the size and the formula. A limited amount, perhaps, about what the formula is, what functional groups are present, but primarily a size indicator, and it'll give us a little bit of ability to distinguish between similar molecules. So as we go forward, we look at a common mass spectrometer. This is one produced by a company called Thermo Scientific, but there are many out there. Basically, we'll see the sort of cartoon schematic of this in a moment, but this particular instrument is equipped as well with an auto sampler that would allow you to run multiple samples over the course of time, so you don't have to stand there and inject each one separately. But all of the sort of functional parts of what we're going to talk about are inside these boxes. So basically, mass spec occurs in a three-step process. And depending on which sort of instrument you're using, which type of sample you are using, you'd have some of these different pieces. But at its core, mass spec requires that molecules be ionized first. And this can be done by a variety of methods. Typically for smaller molecules, like in the organic lab, we'll be talking about EI and ESI. Typically for larger molecules like proteins, maybe in a biochemistry analysis lab, you might talk about MALDI. But in any case, these processes cause the molecule to become ionized. And we'll see that on the next slide. After the molecule is broken apart and charged, then it goes through some sort of a mass analyzer. You can think about this as the part that separates the pieces of the molecule. What mass spec does at its core is that it uses high energy to break apart a molecule to varying degrees depending on what the ionization source is. And then those pieces go through and get separated out by essentially by size, or what we'll see as a mass to charge ratio. This can be done with several different types of instruments, or different types of mass analyzers rather in the instrument. And so you'll often hear these sorts of acronyms tied together. So for instance, a MALDI-TOF spectrometer would be one that ionizes with the matrix-assisted laser and then uses a time-of-flight analyzer. You'll see Qs, sometimes several Qs, one after the other for more advanced instruments, but these are sort of the building blocks. And then at the end, we have some sort of a detector to let us know when the pieces of the molecule reach the detector, and then we can create the output from the instrument and display that, save it, print it, whatever. The ionization step is critically important, and as I mentioned, we typically think about in organic using the EI and ESI methods. Those are fairly aggressive, high energy methods, and what we're doing here is bombarding the sample with a beam of energy of electrons that has a really high energy. And so we talk about something like this, several thousand kilojoules per mole. We know from our earlier studies that the sort of strength of a typical carbon-hydrogen bond is about 400 kilojoules per mole. And so this beam is more than powerful enough to break off an electron to potentially fragment the molecule into pieces by breaking a CC or a CH bond. And so what we'll end up with are several different fragments, one of which is the molecule here that's essentially the entire intact molecule, but now with one electron knocked off from someplace. Typically, lone pairs of electrons will get knocked off, an electron from a lone pair will get knocked off first, most easily. And then if there aren't any of those, we'll be talking about breaking part of a bond, removing one electron from a bond, and generating this cation radical. Notice the plus and dot for the one unpaired electron, the radical part. Since we are using such a high energy, we also will fragment the molecule. Some of the bonds will just literally break, and we'll end up with the radical part, the charged part, radical part going on one of the particular pieces of the molecule. And since that fragmentation is happening randomly, we'll get a collection of molecules that will be analyzed at the end. But the pattern and the intensities of those molecules, the abundance of those molecules, or ions, I suppose, will be indicative of what the molecule looked like to begin with. So we'll see that in a moment. I should go back here just to say that the reason we would use a different kind of ionization for a larger molecule is that we don't necessarily want to fragment a large polypeptide, for instance, a protein, if we're doing some sort of a biochemical analysis, because there would be so many pieces. Proteins are such large molecules that we would end up with just a lot to wade through. So typically we use a lower energy ionization source for large molecules so that we can try to keep more of the pieces intact, have larger fragments to deal with. Here's our basic design for a mass spec instrument. And so if you sort of imagine that picture from the lab that I showed you a few slides ago, we have the inlet part over here. The sample inlet could be literally you sticking a syringe in here and injecting some material into the ionization chamber, or it could be an autosampler where it's robotically controlled. We have the beam going across here, fragmenting those molecules and charging them. Those pieces then get thrown into this mass analyzer, and so they are using a magnet to affect this turn. If you remember from sort of your physics 2 kind of a class, charged particles in a magnetic field will be bent, will attract in one direction or another. And so what we're seeing here is the bulk of molecules going through, or the bulk of material going through, things that are uncharged at this point would basically crash into this back wall. So those fragments that don't have a charge just crash into the back wall and we never see them. The fragments that are charged would bend, and the amount that they would bend would depend on this so-called m/z ratio, the mass to charge ratio. So things that are smaller and more highly charged would bend like this pink line. They'd bend in an extreme way. Sort of average things might bend as this green beam, and this sort of larger and or less charged molecules or ions here would bend along this blue line. And then we have in our detector a slit that allows us to select what we're seeing at any given time. And so by sort of adjusting these parameters, we can scan for a large number of sizes and charges, this m/z ratio. You can get that on the detector, and then that all goes in and gets integrated into a display. A typical output looks like this. So to orient you here, mass spec is always shown with a percent abundance on the y-axis. So going from 0 at the bottom to 100% at the top. Whichever one fragment is the most common, represents the largest abundance, and that sample is pegged at 100%. That thing is called the base peak. And on this particular graph, that represents an m/z, a mass to charge ratio of 29. So it's worth saying here that most of the time, just because of the sort of statistics and probability of this random encounter of an electron being knocked out of a molecule, most of the time we're talking about the charge being 1. We've lost 1 electron. And so for the most part, we can think about this m/z ratio as being a particle with a mass of 29 atomic mass units and a charge of 1. It's not to say that we couldn't occasionally have a charge of 2, for instance, but that'd be a much less likely event. So generally speaking, when we say m/z is 29, we can think about a fragment of mass 29. That tallest peak for each sample is going to be artificially set to 100% relative abundance. That's called our base peak, and that would be this one here. And then we see all of these other peaks represented on the x-axis as that m over z ratio. So smaller m over z fragments are going to be to the left, larger ones to the right. Typical organic molecules are going to have masses, no greater than a couple hundred mass units. And so typically you'll see this x scale that will be 0 to somewhere in the neighborhood of 150, 200 maybe, something like that. There's a couple pieces of terminology on here. The base peak is that one that has that tallest or 100% relative abundance. Everything else is represented as a relative abundance compared to that one. The parent peak is the unfragmented cation. This is what we saw in the ionization slide a couple of slides ago. This is the case where in that particular instance, we just knocked an electron off, but we didn't fragment the molecule otherwise. So for smallish molecules, generally speaking, the highest m over z ratio here that gives a significant peak, that would be our parent peak, and that's the one that we would think of as an unfragmented cation. So essentially, this example is showing us that the molecule we started with probably had a mass of 44 atomic mass units, and then it got fragmented into smaller pieces. Okay. We do see a little tiny blip here after that, higher than that, and that actually is due to isotope variance. So if you remember from Gen Chem and from your isotope discussions back then, most carbon in our organic molecules has a mass of about 12 mass units, but a small fraction of the carbons have a mass of 13. Similarly, the vast majority of hydrogen has a mass of 1. A little tiny bit of hydrogen has a mass 2. The same kind of thing is true for nitrogen and for oxygen and for chlorine and bromine and other atoms. And so when we do happen to have one of those heavy isotopes included, we'll sometimes see a very low abundance peak or maybe a couple of peaks here out at the high end. So this little tiny peak here is primarily due to carbon and hydrogen isotopic events. So what we want to think about is, well, okay, we have this 29 and 44. I already answered this. What are these peaks at, essentially +1? Those are isotope variability sorts of units. But what we want to look at for analyzing a mass spec is to say, what are the fragments here? If we look at this molecule, this output, we see that the parent peak is 44. Now, because we're in organic chemistry, we can say, how many carbons does that represent? So remember, carbon has a mass of nominally 12, and so at most we could have maybe 3 carbons making up this molecule. That would give us a mass of 36. That would leave us 8 mass units to go to get up to 44. And so something like propane would work quite well for this example. And if we look at propane here, C3H8 does have a mass of 44. Its structure would look like this if we were to spell it out. And so we want to think about what fragments are most likely. Remember that occasionally when a molecule gets ionized in that ionization chamber, one of the bonds will break. And we could just lose a hydrogen. Sure, we could break a hydrogen off of a carbon, and that might account for this peak at 43, for instance, if we lost that 1 mass unit. However, what we want to think about for analyzing structure is what would happen if I broke apart these bonds in between the big pieces. So since propane is symmetrical, we only really have to think about 1. But if I were to break this molecule between a methyl group and an ethyl group here, I would expect to get some fragments that had a mass of about 15. And in fact, I do see a 15 peak over here. And the remainder of the molecule from that fragmentation would be about 29. And I see that. That's in fact my most common peak here, my highest abundance. And so basically the presence of these 2 strong peaks at 15 and at 29 tell me that probably this molecule was just a straight 3-carbon chain. I can explain those ionization events pretty well. It's worth mentioning, if you go back to the beginning of this thing, I said that when a molecule gets cleaved, one of these pieces would be a charged cation radical and one would be neutral. So the presence of these two charged pieces on the detection means that they came from different molecules. So essentially, whichever molecule gave me this methyl radical, I didn't see the ethyl part that had been attached to it because it was neutral. And likewise here, I'm seeing the ethyl part. The methyl part that got broken off would have been neutral, and I'm not seeing that. But together, analyzing millions and millions of molecules, I'm seeing both of those peaks. So when you look at a mass spec, try to avoid the idea of explaining every single peak, right? What is this peak right here? Or what is that peak or that peak or that peak? Maybe it's not particularly important. What you want to look at is the big picture. So in this case, a relatively simple molecule, but I would say there's a mass, total mass of 44. That means at most 3 carbons. Let's start there. Let's be simple. Could it be propane? We look at the fragmentation pattern. Yes, I see a big 29 peak. Yes, I see a big 15 peak. It's probably propane. Typically, we're coupling mass spec with some other piece of instrumentation. So IR, for instance. That'll tell us what functional groups are present in a very definitive way. And so we might know at this point, okay, I have a mass of 44, but I know that I have an oxygen, for instance. I know that I have an alcohol group. I know that I have something else. And that would help us refine our analysis. The other part to say here is that while we are thinking about doing this, by hand or by eye, we can certainly distinguish, some molecules from one another when they're relatively simple. But technology has evolved to the point that we can get down to very, very small mass units. And so, as an example here, both pentane and this sort of complex alkene alcohol molecule have the same nominal mass. You and I would add it up and say it's 72 atomic mass units. But if we use all those decimal points that we sort of tell you to ignore in Gen Chem sometimes, we can see that they are different by a couple hundredths of an atomic mass unit. Mass spec can see that. And with the power of having computational analysis and having databases that we can use built into these instruments, although you and I in the class, in the context of sort of casually looking at a mass spec, we can pick out some functional groups and we can sort of figure out some branching structures. The instrument itself has the ability to resolve very closely similar molecules. So our fragmentation pattern is important. In the example we just saw, we had 2 groups here. These are very common groups in organic chemistry. If you see a peak around 15, it's most likely a methyl group that broke off. You can think about a methyl being either at the end of a chain or as a branch. 29, typically an ethyl group breaking off. And so we can use this information to tell us both the molecular mass of the whole molecule and the structure to some extent based on the fragmentation pattern. So a couple things to keep in mind. Sometimes there is no M+ peak. Sometimes there is no molecular ion peak because of excessive fragmentation. If our ionization source is too strong, or our molecule happens to have a structure that breaks down into really stable fragments, we may not see an M+ peak. And that gets more and more likely the bigger a molecule is. So we'll see an example of that here in a moment. We may also not see certain fragments. And so again, avoid the temptation to try to assign every single peak or to try to find a peak to explain every single fragment on your structure, because you may not see some of them because of the particular electronics of the molecule. As we go forward here, here's an example, and I'm giving you the answer. This is a particular molecule here. We can see 5 carbons with the accompanying hydrogens, a highly branched alkane. In this case, I've calculated molecular weight should be 72, but we obviously don't see a 72 peak. Why is that? Well, this is one of those examples of a very stable fragmentation pattern. What I'm seeing here is if I broke any one of these 4 bonds, I would get the same outcome, right? I would take off a 15 molecular weight unit. So 72 minus 15 gets us down to 57. And in fact, I see that is my most common peak here. So I don't see the actual parent peak because I have a very common way to make a stable This would be a tertiary ion, tertiary carbocation, a very stable fragment. And so basically all of the molecules, or all but an undetectable amount, fragment in that way. And so I see this kind of fragmentation pattern. So basically, this would be a tough one for us if we didn't know the answer already. To say, well, it appears that the molecular weight is 57, we would head down a sort of a bad direction, not knowing that the molecular weight should be 72. And I only put this one up as an example. It would be a particularly challenging one for you to just get with no additional information. But this is an example of where that parent peak might not be present. Here's another molecule. So perhaps the exercise in the textbook says a hydrocarbon gives the mass spec reading below, determine its structure. Okay, so the language tells us it's a hydrocarbon. So we don't have to worry about oxygens or nitrogens or chlorines or anything else. We're just talking about hydrogen and carbon. So we want to think about here, how many carbons could I fit into this 86? All right, so given that carbons are nominally 12s, I could potentially get up to 7 of those. That would give me a mass of 84. But if you try to draw that out, you'll notice that you don't have enough mass left to put hydrogens everywhere that hydrogens would have to be to satisfy carbon's octet rule. So if we back off a little bit, we say, well, maybe I have 6 carbons. 6 carbons at 12 apiece gives me 72. That leaves me 14 mass units for hydrogen, so C6H14. And guess what? That's an alkane structure. That works. So now we might think, okay, I have an isomer of hexane. I've got C6H14 is my structure. Well, which isomer of hexane am I talking about? Do I have 6 carbons in a row? Do I have 5 carbons in a row with a methyl branch somewhere? Do I have 2 methyl branches? What's going on? And so I want to look at this structure and think about what's happening. And I want to point out to you here this sort of repetitive pattern that we can see. We go from 86 down to 71. Maybe that's 72, I'm not sure. But we go from mid-80s down to roughly 70 here. So that decrease of about 15 mass units. We go from there to 57, another 15 or so mass units, or 29 from the molecular ion peak. Then we see another decrease of that 14, 15 kind of a size, and another decrease of the same. So on and on and on like this, one after the other after the other. We see this regular pattern. That's indicative of being a straight chain. Essentially, you can think about this molecule, if it's 6 carbons in a row with all the accompanying hydrogens, getting sort of clipped off. Well, did I break between carbon 1 and 2? Then I would get this peak and a 15 that I happen not to see here. If I broke between carbons 2 and 3, I would get this peak plus this peak. If I broke between carbons 3 and 4, I would get 2 peaks the same at half the molecular weight each. And so I sort of see that straight kind of progressive change in the molecule. It's indicative of a straight chain hydrocarbon. If I had extensive, say, methyl branching, for instance, maybe I've got a 4-carbon parent chain with a couple of methyl branches, I would be more likely to see a 15 here because those methyl branches could break off to give me a 15 fragment, as well as the ends of the parent chain. I don't see that here. So certainly in order to have made this little peak, I would have had to have had a small peak over here. I'm just not seeing it. And that's okay because I'm not going to fall into the trap of trying to analyze every single peak. So here's what it would look like if I did have straight chain hexane, just normal hexane. An ionization of the whole molecule gives me the M+ peak at 86. And then fragmentation again into just losing a methyl, losing 2 carbons, losing 3 carbons, or losing 4 carbons. And those are the sort of peak groups that I saw on my analysis. Now again, I have all these little tiny other peaks here that could be due to who knows what, lots of little molecular analyses. Remember, we're blasting this thing with a really highly powerful ionization source. So all sorts of unlikely rearrangements and things could happen. But my pattern indicates that, in fact, I do match straight chain hexane. So that'd be my analysis for this one. Thankfully for you and I, we're not 50 years ago, we're not 30 years ago, we're not even 10 years ago. We have computer analysis. So that mass spec that you're loading your sample into is tied to a computer that maybe is tied to the internet, or maybe it's a local database, but the databases that are out there put together by chemical synthesis companies, analysis companies, forensics, all sorts of different databases have hundreds of thousands of entries, and this number is probably outdated. And we can do something called molecular fingerprinting. So basically, all of those little tiny peaks that you and I can't necessarily make sense of, those will be common. If the same molecule undergoes the same fragmentation, it'll give the same fragmentation pattern. And so we can find those, we can match those computationally and get a really high degree of confidence in our identification, our analysis. So as we go forward here, for us, for textbook examples, for simple things in the lab, we're going to be interpreting mass spec data by locating that M plus peak, probably as the molecular mass, unless we're dealing with a very large molecule or a particularly stable fragment. And then we're looking at mass differences between major peaks. And so a couple quick examples. One of these spectra is for methyl cyclohexane and one for ethyl cyclopentane. They have the same formula, the same molecular weight. So which one is which? Well, we want to look at the differences. Notice that even though they have the same groupings here, same sort of pieces, I see that in this case, going from the molecular ion peak at 98 down to this peak at 83, or that change of 15 units, is really, really common. So I'm losing a methyl group, and that's really, really common. Well, how is that going to work? Probably breaking off the methyl group from the cyclohexane. That makes more sense than just breaking off one of the two carbons off of the ring here. So we can use subtle differences like that to say, what's the most common thing here? The top one very likely loses an ethyl group to get to this to this base peak, and this bottom sample B loses a methyl instead. So this is pretty indicative that I have a methyl branch, and the other one pretty indicative that I have an ethyl branch. Similarly, if I look at these two, again, two isomers that have exactly the same molecular weight, very similar. Well, what's our base peak again? So 2-methyl-2-pentene, so a 5-carbon alkene with a branch, compared to a 6-carbon alkene. So let's look at it. Well, one of them is very likely to have lost a 15 molecular weight unit to go from the M plus peak to this one at 59. That probably tells us we had a methyl branch compared to the other one that didn't do that. There are some common patterns we can look at. In addition to hydrocarbons, alcohols, amines, and carbonyl groups, all three undergo something called alpha cleavage. And I'll show you what this means on the next couple of slides. Of particular use to us are these two additional patterns. So alcohols can undergo dehydration, where we're literally losing a water molecule. This is sort of a like an elimination reaction that you would have learned back in the alkenes chapter, where we lose an OH from one carbon and a hydrogen from a neighboring carbon. So we get a mass loss of 18 units. That's particularly useful for identifying alcohols. And carbonyls can undergo this named rearrangement that I'll show you in a moment. So here's an alcohol, generic alcohol. And what's shown here is very generic at the alcohol carbon. But what alpha cleavage does is it's basically a common fragmentation pattern that says we can break the bond next to the alcohol-containing carbon. And so if this were a tertiary alcohol and I had a couple more R groups down here, I could have thought about breaking either of these, any of these three bonds. The way it's shown, we're just showing that we're breaking off this alcohol carbon from the rest of the chain. And so we lose some fragment. Of course, that depends on what this R group is. But we're left with a particularly stable carbonyl alcohol kind of a radical, which is why this happens in the first place. Dehydration is shown there very much like an elimination reaction that you're used to from previous chapters, just in this case, sort of instigated by a high energy blast. Amines also do alpha cleavage, essentially identical to what you saw in the alcohol slide a moment ago. Because this R group has a variable size, depending on what the R is, we're not looking for a particular change in the size, but what we can do is look at a prospective molecule and say, do I see the alpha cleavage products, right? I think this is the starting molecule. If I cleaved it here, if I did alpha cleavage, do I see those two pieces? Likewise, if I cleaved it here or if I cleaved it here, do I see those pieces in my structure? Carbonyls do that as well. All right. Again, we can cleave either of these two bonds. So this, what's shown as an R, could be R or R prime. We'll see both of those, both cleavage events. But we're generating a very stable charged particle here. The McLafferty rearrangement only is relevant if the carbonyl has a hydrogen on the third carbon away. So looking at our generic structure here, there's our carbonyl carbon. This could be an aldehyde or a ketone. I go 1, 2, 3 carbons away. This carbon has to have at least one hydrogen. Why is that important? Well, because it lets me form this sort of pseudo six-membered ring geometry. And that allows this sort of simultaneous shuffling of electrons, whereby the double bond from the oxygen can grab onto a hydrogen from that third carbon away. Its bonding electrons can go in to make a double bond here, and then break that off from breaking a sigma bond here, pretty unusual, breaking that off into two fragments. So what I get from a McLafferty rearrangement is an alkene from the carbon that was 3 away and the carbon that was 2 away, all right, here. And I get now this enol structure that used to be the carbonyl. So again, because the sizes are variable, this isn't super helpful in looking for a particular change in the fragmentation, but I can see these two fragments and sort of lend support to a potential structure I think might be right. It's very helpful when you look at McLafferty rearrangements to draw the molecule in this sort of hexagonal structure and then draw these arrows so that you can see what's going on and you can see exactly which bond will break. So here are some pieces, right? Here's a spectrum from this alcohol. So identifying the fragments. Well, if we add all of this up, 1, 2, 3, 4, 5, 6 carbons, that gets me to 72. The oxygen puts me already higher than this. I'm not actually seeing the M+ peak, but what I am seeing is the dehydration and alpha cleavage products. And if I look at that in sort of an alpha cleavage standpoint, here's a structural view of the same molecule. I could cleave the alpha bond to the left. That would give me a 43 loss and a 59 left over. So yes, I see 43 and I see 59, pretty prevalent. If I cleaved the other side, the alpha, I would get a 29 fragment that's lost, this ethyl group here, and I do see a 29 right there. And what's left would be 73 that I see right there. So I'm not actually seeing the total mass of 102. And in fact, I'm not even seeing the dehydration product, right? That would be 18 smaller than this, so 84. And I'm not seeing that here. And that's okay, right? Remember that we don't necessarily have to see every single fragment. But if we knew that this was an alcohol to begin with, and maybe we knew its formula, which would be a sort of a typical textbook question thing, right? Here's a formula, it's an alcohol, what is its structure? Knowing that we have these particular fragments tells us that the alcohol was on carbon 3. If we had redrawn this with the alcohol on carbon 2 or on carbon 4 or 1 or whatever, we would get different cleavage patterns. And so this spectrum matches up with this structure. So just quickly here at the end, I mentioned that for larger and biological molecules that we often use a gentle ionization method. So something that's not going to fragment the molecule into a billion pieces. And of course, our masses are going to be higher, right? So I mentioned MALDI-TOF as a common example of a protein mass spec technique. This gets us up to about 100,000 mass units. or what biochemists would call 100 kilodaltons. So we just call an AMU a Dalton in biochemistry. So 100 kilodaltons, that's a pretty good size protein. There are bigger, there are smaller, but this kind of technique works pretty well for that. And what this looks like is the same kind of a format. We have relative intensity on the Y with the sort of molecular ion peak, the base peak tagged here at 100%. We've got mass increasing m over z. The problem is that these molecules are so much more complicated. And so it's a little bit harder to read. It's a little bit noisier. But again, with computational help, we can do pretty well. What we're seeing here, this would be the protein's mass, 14,000 or so, 14 kilodaltons. And what we often see are polymerization artifacts where two of those fragments might stick together in the course of going through the instrument. So this would be a dimerization. It's practically exactly double the protein mass peak. And then we see some fragmentation down here, smaller pieces, one in which here halfway down that seems to be particularly stable. We can go more crazy with this. There are entire facilities dedicated to using mass spec in really interesting ways. This particular technique is called SILAC. And you can look that up and sort of read about it. But basically what this one does is that it grows cells. So these are Petri dishes here. It grows cells in several different states. So in this particular picture, we're talking about using light lysine and arginine. So lysine and arginine that have the lighter isotopes of their nitrogens and oxygens and things like that. We have an intermediate mass where we have a little bit heavier lysine, a little bit heavier arginine. And then we have a heaviest state where the lysine and arginine that we've chosen to feed to the cells are imbued with heavy isotopes. And we can buy those reagents from chemical supply companies. So we have one state where we've provided the cells with the lightweight version of these amino acids, one middle, and one high weight. And we can grow those cells up. They incorporate these amino acids just like normal because isotopes don't have any effect on the chemistry of the atoms and molecules. We mix those lysates together and then we can analyze the products. And we can calculate ratios here between how much of the particular protein might be produced from the light cells, the middle cells, and the heavy cells. And we can do some comparisons here. So imagine that these cells are maybe three different kinds of cells. That's not exactly what's shown in this picture, but maybe we have normal cells, maybe we have precancerous cells, and maybe we have malignant cancer cells. And we're seeing a change like this. Well, maybe this tells us now that whatever protein is not produced very much in the light in the normal cells is produced to a high extent in the cancerous cells. Maybe this protein is of interest to us in figuring out why the cells became cancerous. This could be different nutritional conditions. It could be different cancer states. It could be all sorts of different things. But using the relative abundance of our peaks and the fact that our instrumentation can calculate to a very high degree of precision now what the ratios are, we can use that in some very creative ways. So this was taken out of a somewhat older journal, but I thought it really nicely explained the process. If you wanted to check out that reference, or just Google SILAC and look at mass spec analysis like this, and you'll find some really cool stuff. So that's it for mass spec. It's a tricky thing to deal with. Remember that with mass spec, what you're looking for is basically what's the size and what's the formula. We can do some rudimentary functional group analysis with mass spec by looking for some certain patterns, but it's not great. In another discussion, we will pick up and look at infrared spectroscopy, which really will help us figure out which functional groups are present in the molecule. So that's for another day. We're going to stop here. Get some practice with mass spec, look at the examples, read through the textbook, and reach out if you have any questions. Good luck.